Our next speaker is Professor Christina Orengo from University College London. And Professor Orengo has been a leader in understanding how to classify proteins, but also beyond that, the relationship between protein structure and biological function, biochemical function particularly, and understanding relation between structure and evolution. So the title of her talk is Enriching the Evolutionary and Functional Insights Enabled by Structural Data from the PDB. Thank you.
Thank you, Guy. Thank you for the nice introduction, and thank you very much for inviting me. And I just want to start by congratulating the Protein Data Bank on celebrating their 50 years, and also to thank the people involved in that, Helen and Stephen and others, but also to thank the structural biologists for bringing so many amazing structures in that have helped the community and certainly helped the evolutionary community. What I'm going to talk about today is how we've used the structural data in the Protein Data Bank and some new techniques we've developed to bring extra structural information into the Cas superfamilies and perhaps predicted structural information as well. And more recently, looking at the AlphaFold developments and Rosetta Fold and seeing that AlphaFold were going to deposit so many protein models into the EBI, we wanted to start exploring what that new data, how that helps us to expand our families. Can we find any new evolutionary families? How much structural novelty does that data really bring? Just to summarize, over the last 10 years, although we've had increasing numbers of structural domains being brought into CAF, you can see that the number of evolutionary families has sort of stabilized at about 6,000 and the number of folds at about 1,400. So we're really curious to see, will we find some more with the AlphaFold data? And for those of you not so familiar, this is a classification. We start with structural information because we can, it's much easier to go far back in evolution. The structure is much more highly conserved. And you always get this, you can see here in blue, this very, very highly conserved structural core, even between remote homologs and paralogs. And currently we identify about 6,000 of these families. We use a range of techniques. We have our own structural comparison methods, but we combine that with other people's methods like TM-Align, also with sequence-based approaches. So we have machine learning tools, much more simple than the ones you heard from John this morning, but still quite effective in finding homologs. Once we have enough structural information in a family, we can use the sequences of the relatives to look for patterns and profiles that are a signature of the family. That allows us to find, for example, positions that are very highly conserved. And this pattern of conservation can be captured in a hidden Markov model. And that means that we can take all the sequences that don't have a structure in UniProt and predict whether they might belong to our Cas families or not, whether they match the HMMs. And that way we can expand Cas from about 500,000 domain structures to 150 predicted structures from the sequences. And this is a 300-fold expansion in the data, and useful because it brings in a lot of extra functional information as well. It allows us to understand how these families have diverged in their structures and functions. And in fact, we can see that some of the families have diverged a lot. About 1% of the families, about 50-odd superfamilies, occupy the domains within them are nearly half of Cas. These are very, very highly populated, and they also tend to be quite structurally divergent as well. And the structure divergence often correlates with functional divergence. So we often, with our superfamilies, we subclassify into what we call core structural clusters, where relatives are superimposed within 5 angstroms. But we also try to subclassify according to functional groups as well, because we want to understand how functions diverge across the superfamily. And for this, obviously it would be ideal if you had the experimental information about all the proteins, but we're talking about millions of proteins. And in fact, we really only have experimental functional data for about 1% of the proteins. So we can't use the experimental data. The structural data is actually too sparse. And so we have to use the sequence information. There's 300 times more of it. We can cluster together very highly similar sequences. And again, once we have enough information, we can look at the patterns within the functional clusters. So just to illustrate very briefly the concept, in the top here, you have the alignment of this functional cluster and below the alignment of the other functional cluster at structurally equivalent positions, we can see that some positions are conserved across the whole superfamily. These will be important for the folding, the stability, the packing, but at other positions, whether the position might be conserved, but it will be conserved in different ways. And it's these shifts in residue that indicate shifts in function. And it's this information that allows us to segregate the functional clusters within our superfamilies. And we find about 220,000 of them at the moment. Not all of them have structural relatives. As I told you, there's much more sequence data than structural data, but some of them have structures. And obviously, we were interested whether AlphaFold could help us populate these functional clusters within our evolutionary superfamilies with model data. I should say that this way of organizing the data, the functional families are structurally very coherent. Form follows function. And even very distant, very low sequence identity relatives will superimpose with something like 2 angstroms within a functional family, compared to about 5 angstroms or even higher between some pairs. So as I've said, some superfamilies can be incredibly structurally diverse. What we've been doing, I should also say functional purity. I won't say too much about that. CAFA is a competition or assessment, a bit like CASP, judging how well you do in functional annotation or function prediction, and you need a low score. And really, I'm just showing you this because we've done quite well in these competitions over the last 4 rounds of them, and we've come in the sort of top 3 for the last couple of rounds. But there's still a long way to go. We still need to improve these functional clusters. We've been trying to do that with the help of Burkhard Rost. So he's in Technical University of Munich, and we've been working with Michael and Maria from his team. And he's been developing machine learning algorithms sort of related to what John was talking about this morning, except they don't use the sequence alignments, multiple sequence alignments. They just use the pool of sequences. And they have techniques that allow you to transform those sequences into vectors, which capture features that somehow capture the structural information. I'm not an expert in these areas. To me, it's something like magic or black box, but they seem to be very effective. You saw how effective the methods that John talked about this morning were for constructing a fold. This is really, we are using these to look for similarity between protein sequences. So what we can do is take the transform sequence, the embedding of the sequence, and we could see that actually that gave an indication of whether two proteins were likely to be functionally similar or not. So we could see that the embedding distances within a superfamily are higher. In other words, there's more difference between superfamily relatives than there are between functional family relatives. So this embedding data is capturing something about the structural features within functional families and also within evolutionary superfamilies as well. And we've used this approach to also generate or develop a predictor for remote homologs to pull these into CAS. We call it CAS-E, for CAS embeddings. And this is just, I'm not going to go into too many details. We actually used a very simple machine learning strategy, an artificial neural network. But you can see the performance is very much higher than BLAST. And also it's higher than hidden Markov models that I told you about at the beginning. And I'll illustrate that as the talk goes on. So this gave us a strategy for pulling even more sequences into CAS. And so we were very excited when we heard about AlphaFold and also RosettaFold, and that we could use this structural information perhaps to endorse these additional evolutionary relatives that we were trying to bring into CAS, but also to try to find new superfamilies as well and new folds. So how do we do that? We took all the data that was very kindly provided by DeepMind and by the EBI. Many of the proteins look like this, a bit like a bowl of spaghetti. You don't really know where the domains lie. You can take these structures. Most proteins have 2 or 3 domains. You can take these protein structures. You can scan them against all the structures in CAS, but it takes a long time. Structure comparison is quite slow. Not only that, but the structural data is very sparse. So if you have very remote homologs, you can't find the boundaries. You can't get very good matches to the domains in some case. So again, we fell back on the sequence data. As John mentioned this morning, it's just so vast, and it really helps you to capture domain relationships. What we did was we took the UniProt sequences and we used our hidden Markov model libraries, We have them for Cas, but we also have them for PFAM. So we first of all scan against Cas because we know the domain boundaries are much better resolved in Cas because we've used structural information to identify them. So they give us much better domain matches. Once we have the Cas hits laid onto the sequence, we can then explore what are the best PFAM matches we get that could fill up this sequence. And we have an algorithm that optimizes the coverage of the sequence. And this allows us to then go quite quickly back to that sort of pile of spaghetti and work out where the domains lie. And once you know where they lie, it all makes a lot more sense. You can see these sort of immunoglobulin-like folds that belong to Cas superfamilies, but you can also see these other globular regions that belong to a PFAM family for which we have no structure at all at the moment. So these are PFAM matches where these families, we either use Cas of known structure or we use PFAM families with no structure. And also we find these matches as well. These regions don't belong to Cas or PFAM. We call them new FAM. So we were able to take and when I say we haven't done this for all the organisms, we've identified all the domains in all the organisms, we've chopped them all out, but we haven't processed them all yet. And I'll explain what I mean as I go on. So we're mostly interested with these yellow ones. We know that they belong to Cas. We have a very good HMM match. We know the structure that seeded that HMM. We can get a very good structural match, and we can pull these in very quickly. We're very interested in these green and blue ones. Are these new structural families? Are these new folds? Or are these extremely remote homologs of what we have already? So we take the sequences for these unknown ones, and we run them through our new predictor, the Cas-E predictor that uses sequence embedding data. And we know that performs very well. It's got something like 90% accuracy. So we can then identify quite well what the superfamily is likely to be. Once we've done that, instead of having to search the whole of the structure database that we have, we only need to search that particular superfamily. And we use 3 algorithms to do that in order of speed. Fold-seek is a new method, extremely fast. TM-Align is not quite so fast, but a bit more sensitive. And SAP is very slow, but the most sensitive of them all. And then we can see if we have an embedding match and a structure match, how many can we pull into Cas? How many are completely novel superfamilies? And I just want to thank Martin Steinegger, who helped us use Fold-seek and benchmark it. I'm not going to explain the algorithm. It builds on methods that he's worked with Johannes Soding and Michael van Kempen, using MMSeq2 and bringing in structural data as well. We benchmarked it, or rather Martin and his team very kindly benchmarked it against TM-Align so that we could see it was very good at recognizing the correct superfamily. As I said, it's not quite as sensitive as TM-Align, and TM-Align is not quite as sensitive as SAP, and that's why we run them all 3. So what do we find? How many of these green and blue PFAM and new FAM domains can we pull into Cas? So what I'm showing you here is all the domains that we chop out of the human proteome, as it were, the uniprot sequences in human, and we can see that a large proportion of human, you do have an experimental structure already. In blue, I'm showing you the ones that we could match with our HMM patterns. So it's quite a lot that can come in that way. And then this paler blue are the ones that we can match using our embedding strategies and endorsing that with a structural similarity based on the AlphaFold model as well. So we can be very confident that these are within Cas. For these ones, for Cas-E, we can see that we're getting a very good Cas-E match, but some of the structure similarity matches are sort of just below the threshold, and we will need to do manual curation of those. And that's why we've always done manual curation for these extremely remote homologs. And so we believe these do belong to Cas, but we need to do some more curation. We haven't had time to do that. We've only had this data for about 5 weeks. And this is really about 90% of the human domains we can now pull into Cas, which to me is interesting because these superfamilies stabilized about 10 years ago, and the structural biologists did a really good job, I think, of sampling all the evolutionary superfamilies, and I think we had representatives for a long time. But what about these new ones, these gray ones? What can we say about these? I'll come back to that in a minute. I just wanted to quickly show you another organism. This is, I'm just showing you the model quality that we get for this coverage. Again, the red is known structures, and here the intensity is the model quality. So dark is a very good quality. So anything above 70 is considered good enough to do analysis with. And so there's quite a lot that are above 70. You can see ones that we'd already recognized by HMM. The new ones that we bring in, again, there's a reasonable number above 70, the dark green and the middle green, but there are still some that where the model quality isn't quite so good. And just to show you a similar plot for Leishmania, we chose this because it's a nasty parasite that affects many people in tropical countries, and it can be lethal. And you can see really not many structures in the PDB at all. And yet there's a lot of good quality models that come in from AlphaFold. And also for the ones that we've recently recognized, these CAS-E ones that we recently pulled into CAS. Again, quite a lot of good models. So far, we've only completed this analysis for 3 organisms, and that's because the SAP scans are very slow, but they're very important for bringing in these extremely remote homologs. And we are doing manual curation as well, so we don't expect to finish this before Christmas. But you can see for us some interesting trends. Whilst the coverage of the model quality is similar, we can see that when we look at the CAS families, the ones that we'd already recognized, which I'm showing in blue, these tend to have a higher proportion of good quality models than those that we've brought in, the recent ones that we brought in from PFAM and NewFAM. So the recent ones are in green, and there's a somewhat lower proportion of good quality models for these new subfamilies that we've brought into our CAS superfamilies. And we were interested why that could be. In fact, when we look at the things that we had in CAS before, we can see that these can be modeled quite easily. These tend to be rather large families, I think. They easily give above the high model quality score. When we look at PFAM, we can see most of them can be modeled reasonably well, but there's increasing numbers that aren't so easy to model. And these NewFAM ones are really, really very difficult to model. We think this is partly the size of the family. When we look at the sizes, without going into this in too much detail, we've subclustered all the superfamilies and put things into bins of different sizes. And what I'm effectively showing you here is all the new ones that we brought in pale blue, all the PFAMs and the NewFAMs that we've brought in. These tend to be in much smaller families. So that's one thing that could be problematic. As John mentioned, they need a certain number of sequences. Also, we think they're very species specific, and that's a problem too, because there won't be so much sequence diversity for that reason. We can also see that they tend to have a higher proportion of disorder. So the CAS domains tend to be, a lot of them, 88,000, have no disorder really, and mostly quite low disorder. When you get to PFAM, you can see more disorder coming in and a fewer proportion with zero disorder. And NewFAM, that's even more prevalent. There's not a complete, this is the correlation with disorder, so it's not completely correlated. So there's a number of reasons why these new things we're bringing in are very much harder to model, and we're doing more research into this. It's sort of fascinating. But the question we had was how many new superfamilies could we find amongst this mystery set of grey that we can't pull into CAS? So the red and the blue can all be pulled into CAS, we think. What about these grey ones? And again, this is a very preliminary analysis because we've only had time to run fold-seq, and we know it's not the most. It's a fantastically fast method. It's blindingly fast, but it's not the most sensitive. So we need to go through all these results again and also complete all our SAP analyses. But we find so far, and we've only analyzed 11 of the model organisms, that we get a few hundred more superfamilies and more fold groups. So it's not a massive expansion. We expect these numbers to reduce actually once we do the SAP scans, but it gives some idea of the increase in new families we can expect to see. I'm just putting a huge health warning here because we haven't done manual curation. We haven't done these very slow sensitive scans. And you can see that some of the models make sense on the left, and they look a bit similar to things we've seen before. So we're not seeing anything hugely unusual. And when we do see strange things, you can see on the right that really, although the average model quality is reasonable, you can see there are regions of the structure that really haven't been very well modeled. So we're going to have to do a lot more digging and a lot more curation to make sense of these and to see whether they genuinely are new superfamilies or new fold groups. I just wanted to say a little bit then, because although it's exciting to find new superfamilies, I think a lot of them will disappear. And I'm actually more interested in how these new models allow us to understand more about the structural clusters and the functional clusters within cath. So these are predicted structural and predicted functional clusters, because we've predicted them from the vast amount of sequence data we have, but for most of them, we didn't have a lot of structural coverage. What do we find? If we look at our functional families, how do we increase the coverage of functional families with the AlphaFold model data? Again, we've not analyzed all of them yet, but we can see for some organisms like human and mouse, there was already quite a lot of good coverage, because these are families, some of these families that cover many species, and there were already structures available. But for some organisms like Arabidopsis and maize and rice, important agricultural crops, for example, we can see up to 7-fold increase in the coverage of our functional families. That to me is tremendously exciting, because it means that we can start to understand how the structure is changing and driving changes in function. We can start to see whether variants in the structure are likely to lead to changes in the function, and I'll come back to that in a bit. We also looked at coverage of our core structural clusters. These aren't so highly correlated with function as the FUNFAMs, but there is some correlation because they're sets of relatives that superimpose within 5 angstroms. And again, we can see a considerable expansion in coverage. I'm just showing the largest superfamilies here. Just again, you can see how we've expanded these with new structural clusters, and also with in terms of good quality models. And for some superfamilies, we're getting something like a 30-fold increase in coverage of these structural clusters. So to me, this data has been tremendously exciting, and our research, we've only been doing this for 5 or 6 weeks. So this health warning, this is all very preliminary, but we can see it's bringing in some new superfamilies. But for me, more exciting, it's giving us coverage of these distinct functional groupings within a superfamily. And that will allow us to use the structural data and understand how function diverges. And just one application, I want to end a couple of slides with this, where we've used AlphaFold and also RosettaFold. This was a collaboration with David Baker's group, which we started, and where we also decided to pull in AlphaFold models once they appeared. And we looked at human proteins which were linked to disease, but they had no structure previously. Nothing had been solved, no homology models. We could map the domains in these, all the domains could be mapped to FUNFAMs, either cath FUNFAMs or PFAM FUNFAMs, where we had good models. And we could see that there were 1,300 odd disease mutations that had never been explained before. Could these models help give a little bit of insight into this? I should say that we could see that for both methods, you get very good quality models for cath again. In fact, these models between the two methods superimpose within 2 angstroms. When you look at the PFAM, it's interesting, again, very good quality models for both. And again, you're still superimposing within 3 angstroms. For some of them, it's not such good quality, and they are more problematic with these smaller, more species-specific PFAMs. But for new FAM, it's very much more problematic. And you can see sometimes AlphaFold has a good model, sometimes RosettaFold is the better model. And so we can choose which we use. But just to show that we, having mapped these onto FUNFAMs, and we have a structural representative now, thanks to AlphaFold or RosettaFold, we used our FUNFAMs to predict the functional sites, because as I've told you, we can, for example, if we look at the top bit here, we know that this FUNFAM will have a lysine in a particular position, or an isoleucine or whatever. We can define the functional sites more accurately using the FUNFAMs. In fact, we found known functional sites are highly enriched in the predicted residues from our FUNFAMs. And this is important. There's very little functional site data. We need DeepMind and David Baker to get onto that. But there are other prediction methods, and we included those as well. And that allowed us then to go back and look at how many of these disease mutations could we say something about now that we had these good quality models from AlphaFold and RosettaFold. And you can see 76% of them, we had some explanation. Either they were in a protein interface or a ligand binding site, or in between, at least sort of these sites overlap sometimes, or they're a very highly conserved site in our FONFAM. And these often coincide with allosteric sites. Where they weren't near binding sites, we could see that they were destabilizing the protein. So we had some rationale for these human proteins. And I think I should stop there. I hope I haven't overrun. I just wanted to show one example from each method, one which is a disease linked to, it's an eye disease where you can see the mutation lying within the predicted ligand binding pocket, and RosettaFold model for a protein linked to epilepsy and diabetes where the mutation is in the interface. So I'll stop there and just summarize that by using these new deep learning strategies, we could bring many, many more relatives into cath, and we could validate those sequence relatives by looking at the AlphaFold models and looking for structural similarity. And we can see that actually the Protein Data Bank knew about this 10 years ago. So these families were identified using PDB data nearly 10 years ago. But we can see, thanks to AlphaFold and RosettaFold, we'll be able to increase the structural coverage, and we have done, of functional families and of structural clusters within our superfamilies. And also I showed you an example of how this new data is helping us to get insights into disease. And so I'll finish very rapidly by thanking my group. And actually the FONFAMs were developed as part of the structural genomics initiatives, the PSI initiatives. And I particularly want to thank the people on the AlphaFold project. Sudat left my group, but he has his own group in Malaysia now. And I won't read out all the names because I don't think there's time, but I do want to say particular thanks to Nico, who actually coordinated this and gave up some of his wedding holiday time to try and help finish it over the summer. And also to thank Burkhard and his team, Michael and Maria for the Kathy predictor, Martin and Stephanie for the help with FoldSeq, and also Samir, who helped us with the data and with some of the metrics and some interesting questions. I'm sorry if I've overrun.
Well, thank you.
Do I stop sharing there?
Yeah, thank you.
Sorry, guys. Should I stop sharing or?
No, you can keep that up, I think.
Okay.
So yeah, it is a little bit over, but we can just do a few questions. Samir Velenikar asked, could we see all the questions? And I asked the Biophysical Society people to tell in the chat how that can be done. But I also say again that all the presentations are recorded, so people can access those later. So there's a few questions about intrinsically ordered structures. How can intrinsically ordered structure be classified into cath? From Matthew McCoy. And Carol Burka, how did intrinsically disordered parts predicted with AlphaFold work with cath? And I have a kind of similar question. When you get to those new families, potentially new families, are you using just the reliability scores to say that they're highly disordered, or there's some other, because those reliability
I probably, thanks, Guy. Yeah, I should have, I probably went past that too fast. We use the MobiDB resource, very nice resource set up by Silvio Tassata. And so we used all their predictions for the disordered regions, and that's extremely useful. Mm-hmm. And as you can see, all the different types of families have disordered regions. These smaller, more species-specific, possibly more irregular structures, we don't know. We need to look at them in more detail, seem to have a bit more disorder. But many of the domains within cath have very important regions of disorder. And I mean, some examples, of course, include around active sites. Quite often, the catalytic residues are in highly disordered regions. So when we bring a whole domain into cath, we bring it in with its ordered regions and with its disordered regions as well. And that's how many of those disordered regions come into cath. We don't remove them, and they're often extremely important for the function. I don't know if that answers your question.
That helps a lot. Yeah. There's another question about defining functional families from Eileen Jaffe. When you're doing this machine learning on functional families, how do you consider moonlighting functions? The fact they have multiple.
That's a very interesting question. Yes. Well, actually, we published something. I can't remember how long ago it was. A few years ago. So Shiny Das, very talented researcher who developed the FONFAMs, also used them to predict moonlighting and did quite well by an assessment of moonlighting prediction methods or whatever. So they can pick up sequence patterns that relate to a different function a protein might have, that's possibly a different site that is used in different circumstances or different environmental contexts. So really, they're not doing anything that magical. They're picking up these highly conserved patterns. And if that's been an important moonlighting function that's been conserved for some reason, then we'll be able to find that pattern and identify the subclassify according to these moonlighting functions as well.
So when you have a learning on structure, you have a loss function that compares coordinates, the model versus the experimental and the training, as John Jumper talked about. What about function? How do you calculate a loss function? How do you know two functions are more or less similar? How do you quantify that?
You need to talk to the CAFA people for that. And there's a lot of debate around that. And it's a much harder assessment, I think, than CASP. And it's still the whole assessment is still evolving because it's very difficult to compare experimental functional annotations. Even we use the gene ontology because it's very comprehensive, and there are ways in which you can compare different parts of the Go ontology tree, if you like, and you can look to see how far apart you are in that tree. There are ways of measuring that, so it might not be the best way of assessing functional similarity, but it's the best. It's the one that's used at the moment, and that group are very interested in new ideas for comparing, for measuring functional similarity. We also look at enzyme classification terms as well. We look to see how well do we predict whether two sequences are likely to have the same enzyme function according to their enzyme classification terms. So there's quite a lot of experimental enzyme information that we can use to validate our methods.
That's great. So there are some more questions in the chat which you'll be able to access and respond to offline. So Thank you very much, Christine. Thanks for participating in this. It's great.